This is the second part of the Chapter 2 lecture notes on prokaryotes. And I didn't get a chance to finish this completely. I almost did, but let's just go back and review the parts of a bacterial cell. And remember, a bacterium must have a nucleoid region. It's always going to have a nucleoid region containing a circular DNA chromosome. And it's always going to have a cell membrane or plasma membrane. It's always going to have a cell wall. And it will always contain ribosomes. But the other structures, the capsule is only sometimes. And the capsule of a bacterium can be slimy consistency to help it stick to surfaces, or it can be more of a solid consistency. And some bacteria don't have capsules. The pili are little hairs that help the bacteria attach to surfaces. Again, some bacteria have those and some don't. And bacteria that have flagella typically are motile bacteria. They typically can swim, can move in their environment. Now, we learned a little bit about the reproduction of prokaryotes. I keep saying bacteria. Don't forget that archaea are also prokaryotes. But we talked about the reproduction. I know we learned that it's called binary fission, which means to split in half, to split in two. So basically, prokaryotes do not undergo mitosis, but the circular chromosome is replicated and the two copies separate from one another. And that is binary fission. So what the prokaryotes do is clone themselves. They literally clone themselves. And this is asexual. Binary fission is asexual reproduction. It doesn't give any opportunity for the bacterium to produce offspring with genetic variety. But there is a way for bacteria to increase genetic variety. One of these ways is transformation. And again, this should be reviewed from Bio 111, but transformation, this was, when we learned this, the scientist that discovered transformation was Frederick Griffith. And he discovered that he was studying mice, and he discovered that the bacteria that killed the mice, if you heat killed the deadly bacteria and mixed it with live non-deadly bacteria, that the chromosome from the deadly bacteria could enter the non-deadly bacteria and cause them to express the deadly gene, and that was called transformation. So this is when the cell takes up the DNA from the environment. The prokaryotic cell takes up DNA directly from the environment. And transduction is when the DNA is, a bacteriophage virus is used as a vector to inject the DNA into the cell. And the bacteriophage's DNA, remember, a bacteriophage is a virus that infects bacteria. So the bacteriophage's DNA contains DNA from another bacterium, okay? So that's transduction. And then conjugation is from one bacterial cell to another. And that is when one bacterial cell, actually it's referred to as the F positive, contains the F positive gene or plasmid. This is the plasmid. And a plasmid is a small circular piece of DNA in addition to the large circular DNA that serves as the bacterial chromosome. So a plasmid is extra DNA. And so one of the cells that contains that plasmid is called F positive, and it will transfer that plasmid containing other genes to the F negative cell. And then after that, the F negative cell becomes F positive because now it's going to have that plasmid in it. Okay. Here's how we classify bacteria and archaea. This is trying to think of the name of it. We learned this. Gosh, it starts with a P. Don't worry about it. But anyway, this is a diagram that classifies the three domains of living things. We have, according to everything that we know in biology, there was a universal ancestor, which was a single-celled prokaryotic organism that gave rise to an organism, a prokaryotic organism that evolved into today's bacteria, domain bacteria, and then another organism, prokaryotic organism, that gave rise to the first eukaryotic organisms and the first archaea. So the archaea and the eukarya, you can tell, are more closely related than the bacteria are to either one of them. And the classifications of archaea are, there are euryarchaeotes, and then there's crinachaeotes, there's nanoarchaeotes, and there's core archaeotes. And then in bacteria, there's the proteobacteria, the chlamydias, the spirochetes, the cyanobacteria, and the gram-positive bacteria. So we're going to talk about the classifications of these prokaryotes. A lot of the differences have to do with the composition of their plasma membranes and the composition of their cell walls. All right, so differences in the phospholipid layer of the plasma membranes between archaea, bacteria, and eukarya. Okay, so the archaea phospholipids differ from bacteria and eukarya in two ways. All right, even though the archaea and the eukarya are more closely related than either is to bacteria, when it comes to phospholipids, the archaea is different from the eukarya. So it's different from both bacteria and eukarya. So first they have branched bitanal side chains instead of linear ones. And this shows you the branched bitanal side chain. Second, an ether bond instead of an ester bond connects the lipid to the glycerol portion. So it's an ether instead of an ester linkage, and this is what an ether linkage looks like. And then some archaeal membranes are lipid monolayers instead of bilayers, so they don't even have a bilayer all the time. Sometimes it's just a monolayer. And they show you the differences here and here. Monolayer. Okay, now I see. It's a cross section. So that's showing a monolayer, just one single layer, and this is showing a double layer. Okay. Cell wall differences between the prokaryotes. The bacterial cell walls contain a chemical called peptidoglycan, and archaeal cell walls do not contain peptidoglycan. They can have a chemical called pseudopeptidoglycan, pseudo meaning false, so it's maybe similar, but it's not the same as peptidoglycan. They can have polysaccharides in their cell walls, kind of like plants contain cellulose, which is a polysaccharide, and fungi contain chitin. I'm sorry, what did fungi? Fungi contain, it's chitin, which is a polysaccharide also. And glycoproteins can be found in their cell walls, and they can have protein-based cell walls, but they do not have true peptidoglycan in their cell walls. Bacteria can be divided into two major groups. This is a lab that is done in most college biology labs based on their gram stain results. Some bacterial cell walls can be stained purple, and they are called gram-positive bacteria. They have a thick cell wall, a thick layer of peptidoglycans. This is the peptidoglycan layer here in a gram-positive bacteria. And that cell wall retains a purple stain called crystal violet. And in gram-negative organisms, when you try to stain them with crystal violet, the crystal violet stain won't take. It just washes off. So you have to use another stain, and that is due to the fact that the polypeptide layer is thin. See, it's much thinner, and it contains more lipopolysaccharides, meaning that there's more lipids within their cell wall. So you have to use a stain for gram-negative bacteria called safranin, and it is red, but it makes the gram-negative appear pink in under the microscope. So you end up with, looking through the microscope, a gram-positive bacteria will appear purple, and a gram-negative bacterium will appear pink. And that can help you start to identify the bacterium. So here's some differences and similarities, comparison, contrast between bacteria and archaea. Good little chart to study. As far as cell type, they're both prokaryotes, meaning they have no nucleus. Cell morphology or cell form is variable. Cell wall, bacteria contains peptidoglycan and archaea does not. The cell membrane type in bacteria is a lipid bilayer, and in archaea can be a bilayer or a monolayer. And there are a few chemical differences in the phospholipid bilayer, even if it does have a lipid bilayer. It's different from the phospholipid bilayer of the bacteria. In the plasma membrane, lipids are fatty acids in bacteria, and they are phytanyl groups in archaea. In prokaryotes, as far as metabolism, remember, is the collection of chemical reactions that occur in a cell. So collectively, all of the molecules required all of the substances required to build all the molecules needed by the cell are called nutrients. And nutrients required in large amounts are macronutrients. I know that macros is the shortened term of it, and people commonly talk about getting their macros because you're supposed to get, a certain amount of carbs, a certain amount of fats, a certain amount of protein. But trace amounts of nutrients are called micronutrients. So a few elements are considered macronutrients, the elements that compose the molecules I just named: carbohydrates, lipids, and proteins. And these are those elements. We learned these in Bio 111, the 6 most common elements of living things. And you can remember them from the acronym SHNOP: carbon, hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Be careful with the P and the S. Phosphorus and sulfur. Now, the way that prokaryotes obtain energy is the first thing we ask is where they obtain their carbon from. Okay, so is the carbon obtained from elsewhere? If the answer is yes, then you ask if the energy is from light. And if the answer is yes, we call them photoheterotrophs. So the first question you ask is where the carbon is obtained from. And the second question that you ask is does the energy come from sunlight? So if carbon is obtained from elsewhere, as far as like from the atmosphere, more than likely, then it's a photoheterotroph. So it gets its energy from light. I'm sorry, not from the atmosphere. I'm sorry. This would be like it gets its energy from light, but it gets its carbon from carbohydrates or food that it eats. It has to consume food, but it still uses light energy. But if the autotroph uses light